Good morning, everyone. I hope you all had a good weekend. Thank you for being on time. Let's get right into it. Today, we are going to finish the cardiovascular system lecture, but first, let's begin with a little bit of review, just to go through some of the stuff that we talked about last class. Uh, these are diagrams that are taken from the study guide. So if you have them with you, let's take a look at them together. First diagram here, label the chambers of the heart, use blue and red to indicate O2 level. So once again, this is an anterior view. You're looking at the heart from the front. So left and right are mirrored image. The right side of the heart is going to be carrying oxygen poor blood. And so we're going to be using blue here. That would be the right atrium, collecting blood from the superior and inferior vena cava. Below the right atrium, we have the right ventricle. Right? And on the other side, it would be carrying oxygen-rich blood. And so we'll use red for that. This is the left atrium. Below that would be the left ventricle. Okay, so the left ventricle actually has the thickest wall, thickest myocardium, because it needs to create enough force to squeeze the blood through the aorta and get it throughout the entire body, right? Compared to the right ventricle, the wall is not as thick because you just have to pump the blood to the lungs, which are nearby. So not as much force is required. Okay, so there you go. That's pretty much it. If you like, you can color them blue on the left. Okay, for oxygen poor, and pink on the right, on the left rather, sorry, for oxygen rich. All right, next here we have another diagram. This one is for the various vessels. So up here, we have the superior vena cava. I'm just using short form to save space. Please spell it out in your notes. At the bottom, we have the inferior vena cava. So the superior one collects blood from the top of the body, whereas the inferior collects blood from the bottom part of the body, returning them to the heart. Over here at the top, we have the aorta. Over here, the blood is going to go through this artery and send it to the lungs, and that would be the pulmonary artery. Of course, you have the left pulmonary artery and then the right one here. But I'm just going to put pulmonary artery right there. After going to the lungs, it's going to return to the heart through the pulmonary veins on both sides. So those are the key vessels associated with the heart. Okay, of course, inside the heart, we have valves that prevents backflow of blood. We want to keep the circulation going in a one-way direction to minimize turbulence and to ensure laminar flow. And so we have a valve separating the right atrium from the right ventricle. That would be the tricuspid valve. Tricuspid valve. Similarly, on the left side, we have the bicuspid bicuspid, or sometimes older textbooks, they call it the mitral valve. Collectively, these valves, they're called the AV valves. AV valves, atrioventricular valves. And last time I told you, right, they operate in sync with each other. They are either both open or both closed at the same time. At the entrance to the pulmonary artery, we have another valve right there. That is the pulmonary valve. And at the entrance to the aorta, you have the aortic valve. Does anybody remember what do we call them collectively? The pulmonary valve and the aortic valve together. Yes, Vivian, that's correct. That is the semilunar valve. Half moon, okay? Semilunar. So semilunar operates in sync with each other, but out of sync with the AVs, and vice versa. Okay, last but not least, we have the blood flow here. You should be pretty familiar with that by now. Superior and inferior vena cava feeds into the right atrium, then it's going to go to the right ventricle through the tricuspid valve, of course. And it's going to go through the pulmonary artery to the lungs. They will come back and enter on the left side of the heart. Okay, it's going to make an arch. And one goes up to the head, becomes the carotid artery. The other ones we know it's just going to go to the right side and the left side of the body. This arches down, and eventually it will come back over there. So that is pretty much everything we talked about last class. Any questions before we move on to today's lesson? I hope everybody is up to speed with the concepts. All right, let me bring up the PowerPoint for today. Next thing we're going to talk about is heart rate. Okay, so heart rate is also called the pulse, which is the number of beats per minute. It depends on the person and how physically fit they are. The resting heart rate is going to be quite different for different people, but generally speaking, anything between 70 to 80 is a reasonable heart rate. Of course, heart rate will change depending on what you're doing at the moment. Right? Resting heart rate is if you're just lying down and you're calm, and we take your pulse. That's what the resting one is. But of course, when you get up or if you're walking around, running around, your heart rate tends to go up, right? So that's heart rate, beats, number of beats per minute. There are two other ones that I want you to know. Stroke volume. Stroke volume is the volume of blood that's being ejected from the ventricles every time it's squeezed. Right? So how much blood comes out from the ventricle when it contracts? Typically, it's about 60 to 80 milliliters per beat. Stroke volume might change, can may change depending on the strength of contraction. So sometimes the heart might squeeze more forcefully than others. Let's say you have adrenaline going through your body, then that tends to cause your heart rate to go up and squeeze harder. Your stroke volume is going to go up. People who are recovering from heart attacks, their heart muscle might be a little bit weaker, and so they are not able to squeeze out as much blood as they used to, so the stroke volume might go down. right? So it's not a fixed number. It's something that can change. Um, Finally, we have something called the cardiac output. Cardiac output is the product of heart rate and stroke volume. So, for example, we can write cardiac output equals to heart rate multiply by stroke volume. So example, let's say someone has a heart rate of 70 beats per minute, okay, and then the stroke volume is 80 milliliters per beat. We can calculate the cardiac output. Cardiac output is going to be 70 beats per minute multiplied by 80 milliliters per beat. And just like fractions in math, you can reduce the beats, cross out the beats. And 70 times 80, it's 5,600 milliliters per minute. Okay? So sometimes you can see this as written as 5.6 liters per minute. Of course, there are 1,000 milliliters in a liter, right? Here, I'll put it on the side in case you're not familiar with that. So that's the cardiac output. And cardiac output is a useful indicator to give us an idea of how healthy the heart is. Right? So you don't want a cardiac output to be too high. If it's too high, it either means that the heart rate is too high or the heart is working too hard. It's squeezing too with too much force, right? And that could increase the stress in the heart, right? Uh, Cardiac output that's too low is probably not a good idea either, because it means your circulation is not going very well, right? And it could affect blood supplies to some of the major organs. So by determining and monitoring someone's cardiac output, uh, let's say post heart attack, that would be useful information to judge whether the person is recovering well. So that is heart rate and with some calculations. You will have some calculation questions on the quiz, but nothing crazy, just simple stuff like that. So bring your calculator. Next, we are going to be looking at cardiac cycle. Okay, does anybody know what this picture is showing? This is an X-ray. This is the rib cage you can see here. And there's a little device here. That's correct, Cheyenne and Michael. This is the pacemaker. Okay? So a pacemaker is needed in some cases when the person is unable to regulate their own heart rate. And let's take a look at how heart rate is normally regulated. So the cardiac cycle includes all the events that happens during one heartbeat. Right? So one heartbeat is basically the atria contract followed by the ventricles contracting. Okay, so top squeeze, bottom squeeze, that's one cardiac cycle. Whenever the heart muscle contracts, we call that systole. And whenever it is relaxing, we call it diastole. Okay, systole and diastole. When you go and measure your blood pressure, there are always two numbers, the higher one and the lower one. They say, normal is 120 over 80, right? So the higher number is the pressure that's measured during contraction. So that's called the systolic pressure. And then the lower number is the one that's measured when the heart is relaxing. And that's called the diastolic pressure. Okay, so systole, diastole, it means contraction and relaxation of the heart muscle. So whenever the heart undergoes systole, it is emptying. The chamber is emptying. And whenever it's undergoing relaxation, diastole, it is filling up with blood. Okay? So Imagine this, the two atria contract systole and it's emptying. Okay, where is the blood emptying into? Well, when the atria contracts, the ventricles at the bottom are relaxing diastole and it's filling up with blood. When it's time for the bottom to squeeze, when it's time for the ventricles to undergo systole, it empties into either the aorta on the left side or empties into pulmonary arteries on the right side. And then at the same time, the two atria on top is going to be filling with blood. Okay, so I'm going to write those down for you later on. But basically, that's what these words mean. There's a question. When it contracts, it pushes it out. When it relaxes, it fills up. Yeah, basically. Okay. Pushes it out of the chambers as opposed to when it's relaxing, it's gonna be receiving blood. So what controls the heart rate? No, heart rate is completely an autonomic process. You don't have to think about the beating of the heart. It just happens on its own. Your autonomic nervous system, something we'll learn later, helps you regulate the heart rate. Okay, you fall asleep, your heart is not gonna like forget to beat or something like that. Okay, so we can't control the heartbeat by thinking about it, which is probably a good thing. But there are things we can do to influence how fast the heart beats. Right? So more on that in a bit. There is a cluster of specialized cells that are located just in the right atrium where the superior vena cava comes in, and this group of cells is called the SA node. SA node. I don't think I have the full name here. SA stands for sinoatrial. Okay, sinoatrial nodes. And it is the natural pacemaker of your heart. Under the influence of your nervous system, it rhythmically sends out these electrical signals. Okay, electrical signals are also called impulses. So every time the electrical signal gets sent out, it spreads throughout the rest of the heart and it causes it to beat. Okay? If the signal goes out faster, then the heart beats faster. If the signal goes out slower, the heart beats slower. Okay? So that's the SA node. Further down, there is another node called the AV node. And the AV node is going to basically relay the signal from the SA node and spread it to the rest of the heart. Now, all this is really hard to visualize. So let's take out the study guide and we're going to do some labeling there. And I'm going to put some description for you as well. And hopefully everything is going to be all right. So that's the diagram we're looking at. Now, like I said, you have to print your notes to come to class so that you can follow along. Okay, so here we go. So just to orient ourselves a little bit, so you're not completely lost. What is this structure here? Anybody? This thing right here. Yeah, that's right. This is the superior vena cava. Okay, very good. And then we have the aorta next to it. But that's not the focus of our discussion here. If we look at the right atrium, and you can see this is where the superior vena cava comes in. This is the opening for the superior vena cava. The blood's going to pour in. This, of course, is the opening for the inferior vena cava. So right here, just above where the superior vena cava comes in to the right atrium, that's where you will have the SA node. Okay? SA node. That's the sinoatrial node. Again, it is going to rhythmically send out electrical impulses, electrical signals, and the signal will spread. Some will spread across, and then the other will spread down. Okay, so this is going to um, generate generates impulses, which means electrical signals. Okay, impulses that spread across the atria. And the general idea is this: when the electrical signal, when the impulse gets to a particular part of the heart, if it's a chamber, then it will squeeze. Now, I'm just going to put a side note here for you. It can be influenced by dot 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 dot. Okay, so what are some things that you can do to affect your heart rate? If I want your heart rate to go up right now, what is one thing that you can do and your heart rate will immediately go up uh within the minute? Okay, sure. Adrenaline. Yeah, exercise. That's right. So influenced by physical activity. Okay, you run up a flight of stairs, you do jumping jacks, you lift something heavy. All these things will now increase the firing rate of the SA node, and then your heart rate is going to go up. So someone mentioned about adrenaline. That's a hormone. Okay? It's influenced by a hormone. When you are in danger, when you're scared, when you're excited, when you are stressed out, your adrenal gland releases adrenaline, which then causes your heart rate to go up. Okay. Yeah, anxiety, very good. So, what else? Anything else? Anybody drinking? Oh yeah, thank you, Kyle. You totally took the answer right out of my mouth. I was gonna ask, anybody drinking anything this morning? Right? Probably got a little bit of caffeine, right, to keep you going. So that's chemicals or medicine. Right? So chemicals like caffeine. Besides caffeine, there's like nicotine. If if people smoke, nicotine, THC, right? So all these things can affect your SA node. There are medicine to strengthen the heart and things like that increase the frequency, slow your heart rate, right? Anyway, I think I get the point across, right? It's not something you can mentally change by thinking about it, but there are many things you can do to influence it, to affect the frequency of the SA node. All right, there's another cluster here. This is called the AV node, atrioventricular node. And the purpose of the AV node is basically to relay the signal to the septum and ventricle. So these impulses that originates from SA node after spreading across the atria, it gets a little bit weaker, okay? And then that's what the AV node is for. It boosts the signal a little bit and then it passes it on. And then the signal is going to go across and like this, and then it's going to go down the septum. You remember the septum is the wall that separates the two halves of the heart, and it's going to go down here and basically come back up. This is not blood that I'm drawing, okay? This is the electrical signal spreading across. And so these things have like different names along the way. I'm not going to burden you with it like too much. We just call them like AV bundles. AV bundle, and then more AV bundle. And finally, these things that are in the walls of the ventricles, that's called the Purkinje fibers. Okay. Spread the impulse, the signal, whatever you want to call it, throughout the ventricle. It's okay. Just let me know if you need me to slow down. Okay, and of course, these things will be posted. So let's take a look at what really happens. All right, I'm gonna write down here. So we have the SA node, and it generates the impulse, the signal. So the signal is gonna spread across the atria, which is what I wrote on top. And at the same time, it's gonna be going to the AV node. Right? So during this uh, period, what are the key things that are happening? Okay, so when the signal gets across the atria will contract. So atria contracts. This is called systole. And the blood, I'm sorry, and at this point, the ventricles are still relaxed, okay? Because the signal hasn't get there yet, right? So the ventricles are not contracting yet. So ventricles are relaxed diastole. So because the atria contract, sorry, um, the blood will empty from the atria down to the ventricles, right? So atria empty, blood into the ventricles. And this happens on both sides at the same time. Now, for this to happen, I know you're busy copying, but listen, for this to happen, which two valves need to be open? For the blood to leave the atria and fill up the ventricles, which two valves need to be opened? That's right, Peace. We have the tricuspid and bicuspid. So AV valves open. Now, as the ventricles are filling up, we don't want the blood to leave the ventricles just yet. We have to wait until the ventricles are completely filled up. So we need to close the exit, right? So to speak, which is why when the AV valves are open, the semilunar valves are closed. Are we together? This is the first half of the cardiac cycle. Just emptying the atria, filling up the ventricle. It's not a really hard squeeze, okay? Just a gentle squeeze is fine because gravity does most of the work. We just need to squeeze hard enough to force the AV valves to open, and then gravity kind of does the rest. All right, so as this whole thing is happening, the signal is spreading to the AV node, and then the AV node give it a little boost. Right? It's like if you have a Wi-Fi extender at home, that's what AV node is. Okay, boost the signal, let it reach further, deeper into the house. So AV node spread it to the AV bundle. It's traveled down the septum and eventually is gonna get to the Purkinje fibers. Right? And by the time it gets to the Purkinje fiber, it kind of goes up on the sides of the ventricles. And now the signal, the impulse is at the ventricles. So this is where the atria will relax. okay, are relaxed. They no longer have the impulse. They finish their squeeze now. It's time to relax. So that's diastole. And are filling up with blood. Remember, anytime it's relaxed, it's going to be filling up with blood. So superior vena cava, inferior vena cava feeds feed the right atrium, whereas the pulmonary veins feed the left side. Okay? That's where the blood's coming from for each side. It's filling up the atria. Now, as the blood are coming in from both sides, you don't want them to fall into the ventricles. Not yet, anyways. You need to wait for the atria to fill up. And so that's why I hope you can see that the AV valves are closed. Okay. Close the floor. Don't let the blood drop down. At the same time, all these things are happening because of the Purkinje fiber conducting the signal to the ventricles. The ventricles contract. Right, so this is systole. Right? Where does the blood go? And empty its content. And empty its contents. The blood, basically. Okay. And of course, the right ventricle empties to the pulmonary vein, whereas the left ventricle empties to the aorta. And that completes the cycle. Right? Once the signal gets to the ventricles, it dissipates itself. It's like, fades off. Then the SA node is going to fire another round, and all this would happen all over again. Okay? It keeps on doing this 24/7, 365 until we die. Are we together? Any burning questions for me?
I do have a question.
Please.
Just in the brackets where you have the superior vena cava, like just what reads after
Oh,
that
feed
in the
feed the
bracket?
right atria. Yeah.
Okay, feet. Okay, thank you.
Yeah. There is another question in the chat. Does the RV, right ventricle, empty into the pulmonary vein or artery? Oh, thank you. You caught a mistake. Excellent. Thank you. Thank you. You are absolutely right. It empties into the pulmonary artery. Very good. Okay, I am going to just wait for you to catch up with the notes, and then we'll keep going. Okay, so that's pretty much everything we talked about. You can read more about it, and here it shows you the spread of the impulses in labeled in sequence. So again, read through it yourself and compare with the notes that you've taken. When the natural pacemaker, when the SA node is not able to um, create signal in a normal way, and there are various reasons for that, which we're not going to go into. But if it fails to create regular heart rates, then the person has what's called arrhythmia. There are different degrees of severity when it comes to arrhythmia, and depending on how bad it is, the intervention would be different. One way to fix the problem would be to implant a pacemaker, which essentially sends out its own signal to override the faulty SA node, and then it helps you regulate your heart rate. Okay. So how do we see the heart rate? Okay, how do we see the signal that's being spread across the heart? Well, some of you might have done this before. It's a non-invasive way to basically monitor these electrical signals. Um, Electrodes are stuck on placed onto the person's body at the various location, and then you can see the readout of the electrical signal on the screen. This is called electrocardiograms or ECG. And this is what a typical normal ECG diagram looks like. Now, the scale is important because you have to know the spacing of these peaks and troughs. That tells you whether they're normal or not. Okay, so let's take a closer look at the various part here. When the SA node generates the impulse, that's registered as a little bump right here. Okay, so this first bump basically corresponds to atrial contraction. Okay. That's when the signal spread across the two atria at the top and it causes them to contract. It's not a very strong signal, and I already explained this to you. You don't need to squeeze very hard. The ventricle is just below it. You just give a gentle squeeze, open the AV valves, and the blood will fall down. Okay? So you don't see a very huge spike in electrical signal. Then it gets to the AV node, right? And the AV node, it's passing it down to the AV bundles. So this is just like waiting for signal. Signal is being passed from SA to AV and then to AV bundle. It's just taking its time. Right? There's a little bit of delay there. And once it gets to the Purkinje fiber, then it creates this very sharp increase in electrical signal. That in turns results in a strong squeeze from the ventricles. Right? So this spike here is ventricular contraction. Okay, and the rest of it, don't worry about it. It's just the heart kind of resetting itself so that it's ready for another round of contraction. Okay. So a doctor or even a nurse sometimes who are trained to read these ECG, they will be able to tell just by looking at it if something is wrong or not. I mean, these are pretty obvious. Like you don't even have to be trained to kind of notice that, the top one right here, that's not right. Right? The heart rates are like too far apart, and that's called bradycardia. You don't have to memorize these names, okay? Bradycardia, brady is too slow. This one is too close together. It's tachycardia, or 130 beats per minute. That's like not good. You need to slow it down. And this here is ventricular fibrillation. Fibrillation just means the heart muscle is like undergoing spasm, if you would, and it's not really contracting at all. And a person undergoing ventricular fibrillation, sometimes on like TV show they call it like VFib, right? And they are gonna die basically, right? So you need to use a defibrillator to help reset the heart. Okay? The defibrillator basically sends an electrical shock throughout the chest cavity, and that shock will sometimes kind of reset your SA node and force it to go back into normal rhythm. Okay. Um, Nowadays at malls, at even restaurants, they have this AED. It's automated. a defibrillator, and you just uh, place the uh, electrodes on the person and press the button, and the machine will do the rest. Okay, so that's ECG. All right, let's do some poll questions and see where you guys are at. You guys did pretty good last class with some of the polls. Let's see if we can continue the trend here. Okay, anyone else? So, cardiac output equals to heart rate multiplied by stroke volume. Heart rate is beats per minute. Stroke volume is milliliters per beat. If you cross out the beat, then cardiac output is milliliters per minute. Okay, how much blood does the heart send out every minute? That's cardiac output. Okay, beats per minute, that's heart rate. Okay. Here is another one. All right, excellent. When you're exercising, your heart rate goes up. Since cardiac output is heart rate multiplied by stroke volume, if heart rate goes up, then your cardiac output will also go up. The answer is A. Okay, next we are going to be talking about blood vessels. Blood vessels. Right? This is real. It's what happens when you take away all the tissues and all the organs and you preserve the blood vessels, and you can see it's basically covering the entire body, right? Because every cell in your body needs oxygen to survive, and the way to get the oxygen is through the blood vessel. So you cannot have a cell that's too far away from a blood vessels, right? It's not going to be able to get enough oxygen that way. So that's why it's not surprising to have blood vessels basically tracing out the entire body when you take everything away. Now, the functions of blood vessels. The obvious one would be to transport blood and everything that's inside blood. Next class, we will be learning about blood and lymphatic system. We'll see what exactly is in blood that makes it so important and vital to our survival. Blood vessels, particularly the capillaries, is where there will be exchange of gases, okay, and nutrients. Now, the exchange of gas is going to be a little bit different depending on whether we are looking at capillaries at the lungs or pulmonary capillaries, or if we are looking at systemic capillaries. At the lungs, O2 enters blood and CO2 leaves blood. Systemic means bodily, right? So systemic capillaries, like at the kidneys, at the legs and whatnot. This is going to be the other way around. O2 leaves blood for the tissue to use, and at the same time, CO2 enters the blood. We learned about cellular respiration last semester. You know your cells are constantly creating carbon dioxide. So those carbon dioxides needs to go back to the blood in the systemic capillaries, and then it will be taken back to the heart, and then it's gonna get carried to the lungs where you breathe them out. And other function of a blood vessel is to regulate blood pressure. Right? The blood vessels can actually change its size. If it gets bigger, then the pressure will drop. If it gets smaller, then the pressure will go up. More on that later. I'll write down for you. And another function that's kind of people not really aware of it is that the blood vessels help direct blood flow to the parts that needs it the most. Okay? Your oxygen demand, your blood demand, it's not the same for every single organ at any given moment. So right now you are assuming you're sitting there watching me talking to you, then your blood might get sent to your digestive system and help, absorb the nutrients from the food that you've eaten. But when you're running around, then you don't need as much blood going to the digestive system. You need it to go to your leg muscles, right? If you're exercising with your arms, and it goes to the arms. So the blood vessels help direct the blood flow, right? So that you can have more going to places that you need them at the moment, and then not so much to places that does not require the blood. So how does it do it? Well, here is a real picture on the left side, and we can see the arteries right here. This guy is an artery. And this one right here is vein. They are not actually red and blue like that. To prepare this slide, they have to apply some dye to it. But what are some differences you can see between the arteries and the veins? This cross section, you take a tube and then you cut it, right? Like here, cut it, and then you're looking at it like that. Size, yeah. So I would say the vein here is bigger. The diameter is bigger. Anything else that you notice? That's right, Peace. The artery has thicker walls, right? Compared to the veins, which is thinner. And that makes sense as we talk more about pressure and things like that. But just keep that in mind. Keep this, picture in mind as we progress through the lecture. All right, so here we go. The let me change the color so it stands out a little bit more. We have the artery here, and the blood is coming through, and it's gonna go through the arterial, which is like a smaller version of the artery, and then we have a capillary here. Okay? So the blood's gonna go through, and then it's gonna go to the vein. Right? So this happens. Now, at the beginning of the capillary, you have a bunch of these sphincters, okay? Sphincters are like valves. So there is a valve here, there's a valve here, valve and valve. If we close off the valve, then the blood is just gonna pass right through. The blood's gonna pass right through and then go to the vein. So that's still supplying oxygen to this region, but not to its maximum capacity. If this location, wherever it is, requires more oxygen, then the blood vessels will open up these sphincters, and then the blood will now be able to fill up the entire capillary. Right? And in doing so, you are supplying more blood to this location. 100% supply, as opposed to before, maybe it was at like, 70% capacity. Right? So by turning on and off the sphincters, you allow more blood or less blood going through the capillary. But you never shut off the whole thing. Okay, you shut off the whole thing, no blood gets there, and then the tissue will die, right? Okay, you need to have some blood, but it's just a matter of how much. Okay, it's just like the faucet, right, at the sink. You can turn it on all the way, or you turn it on a little bit. You control the flow. Of course, all this is done automatically in the body. You don't have to think about it. The body just does it for you. All right, we're gonna take a really big note on the topic called capillary exchange. Yes, go ahead. There's a question.
So I wanted to ask, since the arteries and the veins, they deliver blood, do they also have their own blood vessels? Do they also have their own blood supply?
Oh yeah, that is a great question. They don't really um, have their own supply because they are kind of next to capillaries generally. So when the oxygen leaves the capillaries, they kind of supply, you know, uh, the cells in the immediate vicinity with oxygen, including the vessels as well, if they need any. Okay, good question. Capillary exchange. So like I said, this is a big note. You need at least three different colors. And it's an important concept, not only just for the cardiovascular system, some of the stuff we will talk about later on next week and next class also needs your understanding of capillary exchange. Okay, even all the way when we go to kidneys later on, you kind of have to understand some of the concepts here as well. So here we go. Brand new note. Um, so capillary exchange, and then we're gonna draw a bunch of diagrams like here, diagram, and then explanation, and then we're gonna have a table somewhere. Anyway, just plan your space accordingly, okay? And maybe it's a good idea to reorganize the note after class as well. So here we go, capillary exchange. I'll try to write nicer, okay? So you can read my read my writing. So what is capillary exchange? Basically, it's the movement of substances, of fluid, and substances across the capillary wall. We are going to be looking at three factors that affect capillary exchange. Number one, this is the easiest one, diffusion. Number two, blood pressure. Number three, osmotic pressure. Okay, let's do diffusion first. Number one, diffusion. As you know, diffusion is the movement of substance from high concentration to low concentration. Okay, so we're gonna draw a diagram here. Okay, like I know the capillary is more like a net like that. Okay, but that's like super difficult to draw, right? So I'm just gonna draw it as like a single tube. Right? So this is the capillary. And this side is the arterial side. Sorry. Arterial side. It's just the adjective of artery. It's a side that's connected to the, artery, basically, arterial. And over here, this is the venous side. Venous side. Out here is the tissue. So we're looking at a systemic capillary. So if it's a systemic capillary that we are looking at here, the blood that's coming in from the arterial side, they would be rich in oxygen. So that's what these red dots are. This is O2 rich. So high O2. And in the tissue, because the cells are using up the oxygen, so you would have low O2. It's not that there are none, it's just that it is lower. Low concentration of O2. That's what the square bracket means. It means concentration. So as the blood is going through the capillaries, by diffusion, the oxygen will leave. This is simple diffusion, no energy is required. All right, so afterwards, there will be more oxygen on the outside, and then there will be fewer. Okay? And then it's on the way to leave um, the vessels, the capillaries. So, they're not going to come back in, basically. Now, similarly, we have the carbon dioxide, which I'll use blue. The tissue is generating more carbon dioxide from cellular respiration. And then the arterial blood that's coming in, there is a bit of carbon dioxide. Not a lot, because you breathe them out. You're not going to be able to breathe out all the carbon dioxide. You breathe out quite a bit, but there will still be some. Right? So there's low CO2, and here is high CO2. And of course, it goes from high to low. So afterwards, your blood, is going to have more carbon dioxide. Right? And then this is direction of blood flow. Right? So that's not difficult to understand. At least I hope it's not. That's why the blood turns from red to blue as it passes through the systemic capillary, because you're losing the oxygen to the tissue, and then at the same time, you are picking up carbon dioxide from the tissue. Okay, of course, this is going to be reversed. This is going to be opposite if we are looking at capillaries at the lungs. Okay, so I'm going to give you a moment to catch up. Please let me know if you have any questions. We are good? Now, this is not restricted to just gases, not oxygen and and carbon dioxide, right? This can also be true for nutrients. So like, you might have some glucose here. You don't have to add it if your diagram is getting too busy. Okay, so glucose is being used out there, in the tissue, and then the fresh blood is coming in with some nutrients. So this is glucose. Right? And then the glucose is gonna diffuse out as well. You might have some other waste product in the surrounding that might be coming into the blood, right? So the direction of movement is dependent on the difference between the concentration inside the capillary versus outside. Okay? It's always going to go from high to low. And as long as the substance that we are looking at is able to is small enough to leave the capillary wall, then it will. Right? And it's governed by diffusion. All right, so that's the first one. Can I move on? Anyone still copying? Just give me a quick signal if you need me to leave it on for a little longer. All right. Like I said, I will post this. When you log on, you will see class notes and lecture recording. You just click on that, and that's the notes from last class. And then I will add today's notes after class. All right, number two. Number two, what is the second factor that affects capillary exchange, the movement of fluid and substances across the capillary wall? It is blood pressure. Blood pressure. So blood pressure is created by the force of blood pushing against the capillary, against the blood vessels, we can say, but against the blood vessel. Now, to help you understand the effects of blood pressure on capillary exchange, I want you to visualize this with me, okay? Just imagine this in your head. We have a faucet, okay? Like maybe in the garden or something. And you are going to hook this up to a hose. And hook it up to a very, very long hose. Hose. And then you are going to poke holes along the hose. So like poke a hole here, here. Just poke holes along the way. Like that. All right, with me so far? Just think about this. Poke it up. Poke some holes in the hose. Holes. Like a tongue twister. It's difficult to say. All right, then we're gonna turn on the tap to the max. Like, all the way. Now think about what's going to happen. I hope it's obvious, right? Water is gonna shoot out from the holes. Okay? Water is gonna shoot out. Shoot out. However, as you move further away from the tap, further down the hose, what's gonna happen to the water that's shooting out? like maybe over here, do you expect the same intensity as over here, or do you expect it to be more, expect it to be less? What's going to happen? And why is that? Why is it less? Why is it reduced? Well, it's because the pressure is lower, right? As you move further away from the source, from the supply, the pressure drops and there's not as much pressure to shoot out the water. Okay? So I want you to think of this, but now in terms of what we're talking about. So the tab here is like the heart, and here we have the artery, which is really close to the heart, lots of pressure, high pressure. And then over here we have the veins, okay? And in between, of course, we have the capillaries, right? Okay. So blood pressure, the effects of blood pressure is that. So first of all, it's created by force of blood pushing on the blood vessel. Number two, the effect is that blood pressure, BP, blood pressure pushes fluid out of the capillary. Okay? Not pushing blood out. Yeah, you're not bleeding all over the body, all right? You're pushing the fluid portion of the blood out of the capillary. We'll see that next class. If you separate blood, it's gonna be mostly water. Okay, so that's the fluid portion of the blood, along with you know, various things, ions, nutrients, and whatnot. So blood pressure will be pushing them out. All right. We're gonna draw another diagram. Make this one big. We will put a lot of things on it. Okay, so here we go. I'm gonna say it one more time. Make it big. We will put lots of information on it. So again, this is the capillary. We have the oh, hold on, stop. Okay, before I don't know if you use a pen or pencil, but what I want to do next is this. Okay, the capillary actually is leaky, so I'm gonna put some gaps in it. Okay, put some gaps in it. So if you're doing this with a pen, like you might want to do like dotted lines. Okay, and so there are some gaps here and there. I needed to stop you before you actually draw the whole thing. If you're using pencil, then it's fine, right? Capillaries. So I did not mention this before, but capillary walls, the walls are leaky. Right? That's why that's why fluid can move in and out. Okay? But it's not so leaky that the blood is gonna come out. And we don't want that, all right? Because if the blood's coming out, then we have internal bleeding, and that is bad news. We're just talking about the fluid portion of the blood. Now on this side, it is called the arterial side. Arterial side. Not to be confused with like arterial, right? I know it sounds very similar when I say it, but like this is just the adjective of artery. It's the side that's connected to the artery. That's what it means. Arterial is actually the, smaller version of an artery, right? Anyways, over here we have the venous side. Okay, don't forget the O. It's not like the planet Venus. It's Venus with the O side. All right, so the blood pressure on the arterial side is approximately 30 mmHg. Millimeter mercury is just a unit for measuring pressure. And there are different ways to measure pressure. This is the unit we use for blood pressure. And on the venous side, based on everything we discussed with the analogy and everything, I hope you understand it's going to be lower, right? And that is going to be about 15 mmHg. The numbers don't really matter too much. Like you don't have to memorize them per se, but understand that it's higher on one side and lower on the other side. And so we are going to have more of a push on this side, higher blood pressure, pushing fluid out of the capillary. Okay, and that push will get weaker and weaker as we go on to the other side. Okay, but it's there. It's there. Okay. Pushing fluid out into the tissue. Okay? Outside is tissue. Now, I haven't really put in all the details yet, okay? You don't have to do this. I'm just showing you outside here. That's where the cells are. Okay? That's where the cells are. All these sunny side up eggs. That's the cell. Those are the cells, okay? I'm not going to put them in today. We will later on when we talk about something else. But just know that they're there. Are we okay? All right, we'll come back to this diagram. We're going to add more things to it. But why don't we take a quick break first? I feel like we've been talking for so long. We'll come back at 9:50. How about 9:50? 9:50. And then I'll leave the screen on if you need to copy anything else. See you in a bit. Next factor that affects capillary exchange, that would be the osmotic pressure. So I can keep this diagram. We're going to add more details to it. But let's talk about the third factor quickly. Number 3, osmotic pressure. What is it? This is created by solute concentration difference across the capillary wall. Okay. This next part is a little bit counterintuitive. We talked about this last semester, but let's talk about it again just to make sure that we are all on the same page. Let's say we have 2 chambers, and then we have a membrane, and then we have some like purple particles. Let's make them bigger. And purple particles here. Now, as you can see, they are really big, so they cannot cross the membrane that I've drawn. So let's put some water here. These are solute. Too big to cross the membrane. So my question to you is, if I leave this alone, which way will water move? Okay, I'm gonna set up the poll. I want to see what you all think. Okay, so is it A, to the right, or B, to the left? Which way will water move in this setup? Okay, we are half in the answers here. Half say it's to the right, half say it's to the left. All right, this is an important thing to understand before we can talk more about osmotic pressure. Remember, the purple dots are too big to cross. They are not able to move. Okay, they cannot, the purple balls cannot go to the left like that. Okay, so the only thing that can move is the water. Right, so the water is actually going to move to the right side to help balance out the concentration. Listen, things always try to achieve same concentration on both sides whenever they can. So as water moves to the right, you will dilute the right side, and then the left side is going to get more concentrated, and eventually they might balance out, right? As a way to help you understand this, you can think about putting a spoon of salt into your mouth and holding it, okay? Or even sugar is gonna work, right? If you hold it in your mouth, the salt is basically gonna suck the saliva out of your mouth, right? It's pulling liquid towards the saltier area. That's essentially what's happening here. Do we understand? Okay. For those of you who chose B, have I convinced you that it is A? If not, ask me a question so that you will understand the next part. Right? It's not going to go away if you don't fully understand it. So just ask me something. I'm just going to give more space here. Okay. So in the context of capillary exchange, the osmotic pressure is created by solute concentration difference, and these solute can be electrolytes, mostly Na plus, okay, as well as proteins and even cells, blood cells. Okay? So these things, here, let me rewrite that. Blood cells. These things are typically too big, the proteins and the blood cells, they're too big to leave the capillary walls. Even though it's leaky, but these things are really big, it's not gonna, move. So they are like the purple balls, and they are there to increase the solute concentration. So I'm gonna add another point here. Generally, the concentration, the solute concentration is higher in the blood than in the tissue. Right? So it's like this. In the inside, you have more concentration, higher concentration for the solute compared to the outside. So I ask you again, which way is fluid going to move? into the capillary or out of the capillary? Let's do the poll again. Is it A, into capillary, or B, out of capillary?
Excuse me, sir. Can you repeat the question, please?
Sure. So I'm saying generally speaking, the solute concentration is higher inside the blood, in the capillaries. Okay, so that's what these purple dots are compared to the outside. So based on this information, do you expect fluid to be entering the capillary, going into it, or leaving it? A or B? That's the question. Okay. Anyone else? All right. The correct answer is into the capillaries. Remember, fluid is drawn to location that is saltier, so to speak. Right? More solute, it's gonna cause liquid to be drawn to that area. Right? So it's A, into the capillary. So over here, the overall effects is OP, osmotic pressure, draws fluid into the capillary. So let's go back to the earlier diagram. Once you've finished copying that. Okay, so let's take a look here. On the left side, the osmotic pressure is approximately 20 mmHg. Okay. And over here, OP is also around 20 mmHg. So you see, the osmotic pressure is constant. Whether it's on the arterial side or venous side, it does not change because the solute concentration does not change significantly for the blood as it moves across the capillary. It's not like blood pressure where it drops. So we're gonna draw some arrows here to represent the osmotic pressure drawing fluid into the capillaries. And since on the arterial side, the osmotic pressure is less than BP, we're going to have a shorter arrow. Okay, and it's going to be the same arrow going across. Same arrow going across. So at some point, you will see in the diagram, because the blood pressure is dropping, but the OP is not, at some point, the OP, the osmotic pressure, will be stronger than the blood pressure. Do you see that? The blood pressure and the osmotic pressure basically are opposing forces that work against each other. Blood pressure is pushing fluid out, and on the other hand, OP is bringing them back in. And it's a tug of war between the two forces. Whichever force ends up being stronger is going to have the dominating effect. So on the arterial side over here, you can see, here I'll write down for you. On the arterial side, BP is greater than OP. Therefore, we have net loss of fluid. Okay. The blood pressure is stronger. We're pushing more fluid out than we're able to bring in. And as a result, we have net fluid loss. Again, I have to stress we are not losing blood. It's just the fluid portion, right? The water portion, if you would, that's leaking out. Over here on the venous side, you can see blood pressure is less than OP. Therefore, net fluid, net gain, I guess, net gain of fluid from the perspective of the capillary, of course. So you're going to be able to bring some back in like that. Okay, fluid gain. Do you understand? Please let me know if you have any questions. Now, for those of you who has a keen eye, you might be able to see that the difference on the arterial side is 10. 30 minus 20 is 10. And the difference here is 5 only. Right? In other words, you actually will be losing more fluid than you are able to bring back in. Okay? Here you have 10 units of pressure pushing things out, but you only have 5 units of pressure bringing it back in. So there are some leftover fluid in the tissue that you are not going to be able to bring back in. Right? So over time, that excess fluid, we don't want it to build up in the tissue because that's going to lead to swelling, right? So what happens to it, and why would your body not want to bring back everything? Why leave some in the surrounding? That's something we'll talk about in lecture 2. Okay? But I just want you to, be aware of this seemingly a problem, right, with this setup, and then we're going to address how to deal with the problem in lecture 2. That's it for capillary exchange. You sure you're good? Okay. Time will tell. Let's do a poll question. I have 2 poll questions for you. Okay. If you're not sure, you can always choose E or F. Choose F if you're not sure. That way I know. All right. Anyone else before I close the poll? Okay, so the correct answer is A. On the arterial side, blood pressure is greater than osmotic pressure, causing the net pressure. Net is like when you add them up, okay? When they cancel each other out, the net pressure would be out. If you look at our diagram, you can see the red arrow is longer than the purple one. So overall, it is still pointing outwards, which is why we have the fluid loss, right? It is the opposite on the venous side. Okay, one more question. All right, last call. Anyone else? Okay. The osmotic pressure are represented by the purple arrows here, and you can see they are the same. It's 20 on arterial side, and then it's also 20 on the venous side. It does not change from one side to the other because the solute concentration of the blood remains the same. That's why the answer is C. OP is the same throughout. All right, moving on. Next, back to the PowerPoint, we're going to talk about venous return. Venous return is essentially returning blood to the heart. And there are two main obstacles in achieving that. First of all, if you think about the location of the heart, okay, which is this is the person, and then the heart is going to be over here, which means everything below this line needs to go against gravity before it can return to the heart. Right? So going against gravity is not an easy thing to do. You need to expend energy to do that, right? And number two, by the time the blood gets to the vein, the pressure is very low. And anytime we have a low pressure system, then the fluid that's inside is going to move uh, slower. Right? So not only do we need to climb uphill, but it's not really going too fast. And these two things combined presents a challenge to venous return. And so how do we fix that problem? Well, one way to fix it is to use valves. Right? So in the veins, there are these valves, one-way valves exist in veins, and there are muscles that surrounds the veins. These are skeletal muscles like calf, the muscles in your calf, the muscle in your quads. So every time we walk around, every time we move our muscle, the contraction of the muscle helps squeeze the veins. Okay, the contraction of the muscle helps squeeze the veins, and that boosts the pressure up a little bit, and it helps squirt the blood upwards, basically. Okay, so the blood's going to go up towards the heart. It's not really forceful squeeze, right? The blood's not going to go straight back to the heart. It's going to go up a little bit, and then as soon as your muscle relax, then the blood falls back down. But it doesn't fall back all the way to the bottom because of the valves. Okay? The valves will be able to catch the blood. So next time when you contract your leg muscle again, then the blood is going to go up a little further and a little further. So, eventually it's going to be able to climb back to the heart. Okay, so that's why excessive sitting is not too good for your circulation, because when you're sitting, you're not really moving your leg muscles, right? And that really impairs the ability for the blood to go back to the heart. You might see, people who are on long flights, they just like sleep through the whole thing. At the end of the flight, they may not be able to put the shoe back on, right? It's like your leg has swollen. Right? It's because of all the blood that's pooling, right, at the bottom part of the body, and it causes these pressure to build up. And as we talked about, capillary exchange, if the pressure is higher, then these red arrows are going to become stronger, and now you have even more fluid that's being lost to the tissue, and it just pulls up and cause your feet to swell up, right? Same thing with pregnant women. Later on, in the trimesters, as the baby gets heavier and heavier, all that extra weight is pushing down on the body, and it creates more pressure that interferes with this venous return process, and then the feet swells up a little bit. Okay, I remember my wife; she had to get like new shoes towards the end of the pregnancy. Just the leg was so swollen it won't fit into her usual shoes anymore. Now, is possible for people to have damaged veins in the damaged valves, sorry, damaged valves in the veins. And if the valves are damaged, then the blood is pooling because it's not being returned to the heart, right? And all that pooling of blood can lead to bulging of the veins, and it's called varicose veins. Right? Some of you might have heard that before, or you might have this condition. And one way to fix this is to wear elastic stockings, right? So the stretchy stockings is basically going to help improve circulation, right? Um, to basically empty those veins and help push the blood back to the heart. So that's venous return. Next, blood pressure. We already have a good understanding of blood pressure after the notes on capillary exchange. We know it is the force of the blood pushing on the capillary walls. And ultimately, the source of that pressure came from the contraction of the heart, right? Just like the analogy with the water tap, right? If you turn on the tap, that's where the source of the pressure is. When the heart squeezes, that's where the source of the pressure comes from. And as we know, the blood pressure will drop significantly as we go through the various type of blood vessels. And blood pressure itself is actually dependent on various factors. For example, how much blood you have. Naturally, if you have more blood, so increased blood volume, that translates to more higher blood pressure. So one of the things people take if they have chronic hypertension, if they have long-term high blood pressure, is they take what's called a water pill. Has anybody heard of a water pill? A diuretic, that's what they're called. Water pill, what does it do? Anybody know?
The person passed a lot of fluid, like a lot of urine, to
Yeah,
reduce the
exactly.
amount of.
Very good. Okay. That's exactly it. So the water pill, again, diuretic, that's what they're called. People who are taking these things, they pee a lot. Right? So by peeing more, you are lowering your blood volume, right? And that's going to lower your blood pressure. Blood pressure also depends on stroke volume. If you increase stroke volume, if the heart squeezes harder, that's going to translate to higher blood pressure. Vessel diameter smaller equals to higher BP. Think about squeezing a balloon. When you squeeze a balloon, the pressure goes up inside. Right? When it's bigger, then the pressure drops. Okay. I already mentioned to you, blood pressure is measured in millimeters mercury, mmHg, and there is a systolic value that you measure when the heart contracts versus a diastolic value. That is the one you measure when the heart relaxes. Normal blood pressure typically is 120 over 80, but it really depends on the person. Okay. Like a younger person might have lower blood pressure, right, compared to someone who's, older. But this is like just in general, the textbook value, if you would. All right, I'm gonna show you a few graphs, and they are quite important to remember. This graph is for blood pressure change. On the y-axis, it's the blood pressure, okay, measured in mmHg, and on the x-axis, it is different blood vessels, okay, at the distance from the ventricles, from the left ventricle specifically. So it's not surprising that at the beginning, you can see the blood pressure actually fluctuates. It goes up and down between 120 and 80. When the left ventricle contracts, it goes as high as 120, and then when it relaxes, it goes as low as 80. Okay, so that fluctuates. And as we move into the arteries, the arterioles, you can see the fluctuation becomes less and less obvious, right? By the time you get to the capillaries over here, you can see the blood pressure is no longer dependent on the contraction of the heart. Okay. In other words, it doesn't even matter whether it's contracting or relaxing, you're just going to get the same reading. Okay. And then it's just going to continue to decline as it goes into venules, small veins, and then ultimately drops very, very, very low. Okay. So I was telling you, right? In the notes at the beginning for the capillaries, at the arterial side, it is around 30 mmHg. Afterwards, it is around 15 mmHg, right? And it's just a single number. There's no more fluctuation. You're too far away from the heart to feel the effects of the squeeze and the relaxation. Okay? If I ask you on the test, which vessel has the lowest blood pressure, it is going to be the veins, okay? The veins. The further away you are from the heart, from the left ventricle specifically, the lower the blood pressure will be. Okay, so the vena cava, superior and inferior vena cava, that's where you're going to have the lowest blood pressure. Okay? Two more graphs. Velocity change. Okay, so this is the one that we're focusing on, okay? The velocity one. The other is for cross-sectional area. So velocity is basically speed. It depends on two things. Depends on number one, blood pressure. Number two, diameter. High BP equals faster. Okay? And diameter, this is the one people always get confused. Right? And when you think about it, if you have greater diameter, bigger diameter, is it faster or slower? Don't think about the blood pressure. Just think about the diameter on its own. Okay? If you have a bigger vessel, do you think blood is going to be able to go through it faster or slower? Let's do a poll and see. A for faster, B for slower. Bigger diameter, is it A faster or B slower? If you're having trouble, think of a highway. Are traffic going to be faster with 6 lanes or 2 lanes? Yeah, it's half-half, okay. Uh, anyway, the correct answer is faster. If it's bigger, more lanes, then you have more space to go through, right? So in other words, if the pressure is really high and then the diameter is really big, then it's going to be able to go really, really fast. So let's take a closer look at the different vessels. Over here, we can see at the beginning, the velocity fluctuates a little bit. It goes fast and slow and fast and slow. Okay? That should not be surprising because the velocity is dependent on blood pressure. And since blood pressure fluctuates at the beginning, you would expect velocity to also fluctuate. Okay, so over here for the arteries, they are high pressure, but they are smaller relatively. Remember that picture I showed you earlier with the artery next to the vein, right? You can see the vein was much bigger, right? Let's go back here, quickly show you. Right here, right? You see the vein is much bigger compared to the artery. So artery is high pressure, but it's smaller. On the other hand, for the veins, it is low pressure, but they are bigger. Oops, bigger. So, overall, they work out to be about the same. It's a little bit faster in the arteries than it is in the veins, but, they are comparable. At the capillaries, it is going to be the slowest. Okay, slowest. Okay, need to slow down. The blood needs to slow down for capillary exchange. You don't want the blood to be, whooshing by. You need it to give it some time for the oxygen to go in and the nutrients to go in, out, and so on and so forth, right? Like if it's going by too fast, then you don't have sufficient time to have enough gas exchange, right? But once you get past the capillaries, once the capillaries merge together to form the venules and the veins, then it gets bigger and bigger and bigger, and the speed starts to pick up again. Right? So the slowest is going to be at the capillaries. And superimposed onto this graph is the cross-sectional area. Right? All that means is if you lay out the vessels, which one would cover the most area. Right? So aorta is really big, but we only have one of them, so it doesn't really cover much space, much area. Capillaries, individually, they are very small, but capillaries are all over the body. So collectively, they cover the most area. All right. And then veins, again, they are big, but there are not a lot of, there are only two vena cava, right? So they won't cover as much area as capillary. It's like having $100 bill. Okay, it's the $100 bill is a lot of money, but if you only have one of them versus you have like, you know, $205 bills or something. Okay, $5 bills on their own, it's not a lot, but if you have a lot of them, of the bills, then it adds up, right? That's kind of like what's going on here. We are almost there. Any questions for me? Very good. Hypertension is another word for high blood pressure. There are mechanisms, there are ways in the body that would help bring the blood pressure back down when it gets too high. Your kidney, for example, plays an important role in that. There are some hormones in the body that will help you bring the blood pressure down. We'll learn about these things later on in the course. But when those mechanisms are not enough, then the person will have consistently high blood pressure. And even when they're resting, when they're like just lying down, if their blood pressure is higher than 140/90, that's called hypertension, and it requires medication to control it. Okay, because prolonged periods of high blood pressure is not good for the heart and it's not good for the kidney. Okay, it increases the risk of cardiovascular diseases as well as kidney failure. So there needs to be, like I said, people can take the water pill. There are other medications that can take that you can take to lower your blood pressure. Um, Sometimes it's hereditary, it runs in the family. Other times, diet is going to contribute to high blood pressure. What kind of food should you avoid if you have high blood pressure? What kind of food should you avoid if you have high blood pressure? Okay, yeah, fast food. And what exactly in the fast food that makes it not so good? That's right, salty. Okay, salty. You want to avoid salty food. And here is why. Okay, you don't have to do it to your diagram, just watch. If you eat a lot of salty food, I'm talking about like consistently, right? Not like, just have a bag of chips and then your blood pressure is going to go up significantly. It's not like that, okay? It's like prolonged periods of salty food. All those extra salt, again, don't draw this, just watch. All those extra salt is going to end up in your blood ultimately. Right? And that actually is going to increase your osmotic pressure. Right? So these purple arrows is going to get bigger. right? And it's going to draw more fluid into your blood vessels. So all that extra fluid that's coming into the blood vessel, not extra fluid, but like, all that fluid that's like being retained in the blood vessel, it contributes to your blood volume. Higher blood volume results in higher blood pressure. Right? So that's why you should cut down on your sodium intake if high blood pressure is a concern for you. Okay, just a few quick polls here. I think I have 4 or 5 coming up. Okay, just half a minute, guys, for each. Okay, the correct answer is veins. Remember, blood pressure fluctuates at the beginning, but ultimately it goes down until it is the lowest at the veins. The further away you are from the heart, the lower the blood pressure is going to be. Think of the water hose analogy. Next one. Velocity of blood flow is typically lowest. Yep, the correct answer is capillary. Okay, capillary, you want to slow it down significantly to allow gas exchange to take place. Okay, let's try a harder one here. Slightly harder one. Anyone else? Okay, as I told you, velocity is dependent on 2 things, blood pressure and diameter. The bigger the blood pressure, the higher the blood pressure, the bigger the vessel, the faster it will be. So if you want if you want velocity to be the highest, you want pressure to be high and you want it to be big. So the answer is C. C. Okay, let's take a moment and finish this. You can do it yourself. Oh, I don't know why they're all A's, but doesn't matter. A, B, C, D, E. Yeah, whatever. Just choose whether it's artery or veins, okay? I'll give you a moment to do that, and I'll take it up in a second. Right. Higher blood pressure. It is going to be the artery. Higher blood pressure. It's closest to the heart, right? Closest to the ventricle. Thicker vessel wall. Due to the high blood pressure in the artery, it also needs to have thicker vessel wall to withstand the pressure. Right? If the wall is too thin and the pressure is too high, it's gonna burst. Larger lumen diameter. Lumen just means the inside of a blood vessel. Actually, it's inside of inside of the vessel. But it's a term that can be used to describe the inside of any hollow structure. Okay, later on, we'll learn the lumen of the stomach. It just means the inside of the stomach, right? Yeah. So like if this is the blood vessel, that's the lumen that we're talking about. That's where the blood is gonna be. So which one has a larger diameter? You remember the picture I showed you, right? With the artery and the veins, the vein is the one that has the larger diameter. Contain valves, that would be the veins. It helps with the venous return. Blood has higher velocity in the artery, right? Slightly higher if you think of the graph. And carries blood away from the heart. That would be the artery. All right. The last few slides talks about various cardiovascular diseases. It's usually how it goes with these lectures. Okay, we learn the name, the anatomy, learn what it does, the physiology, and then we learn a little bit of diseases, the pathology. All right, cholesterol. There are two kinds of cholesterol. We have the LDL and then we have the HDL. People usually call them good and bad cholesterol. Do you guys know which one is the good one? Which is the good one, the HDL or the LDL? Yeah, this is the good one, okay? Good one, and then this is the bad one. It's the ratio that actually determines whether you are, at a good place or not. It's not just the absolute amount. So HDL stands for high density lipoproteins, and LDL is low density lipoproteins. As you might know, lipids are not they cannot mix with water, and blood is mostly water. So you cannot have the cholesterol just floating around in the blood on its own. It needs to be carried by these lipoproteins, right? So think of it as like a blob, right? The blob has cholesterol plus the lipoproteins. Okay, lipoproteins. So LDL, low density lipoproteins, means the blob is mostly cholesterol with a bit of protein, and that's like the bad stuff. That's the stuff that can build up in the lining of the arteries, and it causes problem. High density lipoprotein means it's mostly just the protein with a bit of cholesterol in it. That gets to the liver, where you will use them to form bile. More on bile later, but this is the stuff that's stored in the gallbladder, and it helps you break down food. Okay. Uh, if you ever had a really violent episode of vomiting, right? You vomit everything you've eaten, okay? Nothing, no more food comes out, and then you still want to vomit. Sometimes you might have these yellowish, greenish things that are coming out and it tastes bitter. That's the bile, right? That's the bile, right? So more on that later. Right? So HDL is the good cholesterol, LDL is the bad one. When LDL builds up along with other fatty substance in the blood vessels, it creates like a patchy bump there. And I told you last class, blood does not like passing through a rough patch, okay? It wants to go through a smooth surface. Anytime it passes through a rough patch like this, right, it creates turbulence, and then the blood is going to start to clot. And that's exactly what happens. When you have a bit of LDL builds up, then the blood clot is going to form on top of it, and then it makes it even worse, more bumpy, and then you have more blood clot, more inflammation, and then the whole thing just builds up, builds up, builds up, builds up. It's like a snowball effect. And before you know it, the blood vessel is going to be narrowed, which, if you remember from last class, this is called stenosis. Okay, stenosis, which is narrowing of blood vessel. Now, depending on where this is happening, it could be not too bad, or it could be really bad, right? So more on that later. So this build up of I should probably label it for you. Okay, this purple thing is the blood clot. Okay, and then the red thing is like the fatty stuff. Fatty stuff. Okay, the cholesterol, for example. Right, the saturated fats that we talked about last semester. Right, all these things building up. So that leads to the hardening of your blood vessels, and that's called atherosclerosis. Atherosclerosis. Okay. Let me show you a picture of what that looks like. Here is a diagram of it, and here is like the actual thing. On the left, we have a normal cross section of an artery. You can see the thick walls, and the lumen, the inside, is unobstructed. Okay, blood's going to be able to pass through it. Over here, this is from someone who died from probably, actually, I don't know what they died from, okay? But then they have a clot in the blood vessels, okay? So over here, you can see the atherosclerotic plaque, which is just the fatty stuff that I talked about, cholesterol, saturated fatty acids. And then on top of it, that's where the blood clot is forming, sticking to the plaque. And with this huge thing obstructing the blood vessel, the blood flow is going to be obstructed, right? It's not completely blocked off yet, but it's restricted blood flow. Right? We call that ischemia. Right? You don't have to memorize it, but if you want to know, it's called ischemia, reduced blood flow. So where can this happen? It can happen anywhere in the body, any blood vessel, right? But it's particularly concerning if it happens in the coronary artery. You might remember coronary arteries are arteries that supplies the heart muscle with blood, with oxygen. The heart cannot use the blood within its chambers because it's going through with very high pressure and high speed. You do not have enough time to extract the oxygen and nutrients from it. So it relies on the coronary arteries to supply nutrients and oxygen to the heart muscles. If the coronary arteries are narrowed because of the atherosclerosis and the buildup of the plaques, or if it's completely blocked off, then it would lead to heart problems. So these things are called coronary artery diseases, okay, or CAD for short. If it's a partially blocked coronary artery, then it will lead to what's called angina. Does anybody know what angina means? Angina.
Small heart attack.
Okay, small heart attack. That's one way of putting it. What kind of pain are we talking about here? Chest pain, that's right.
Heart pain, heart pains.
That's right, very good. Chest pain, okay? So not full-fledged heart attack, just chest pains, particularly after the person physically exerts themselves. Yeah, if they walk up a flight of stairs, they might have the chest pain, okay? Shortness of breath. Lifting something heavy, that might create the chest pain, right? It's because when you do these things, when you exert yourself, your heart needs to beat faster to supply that oxygen. But if the heart vessels are blocked, then the muscle themselves are not getting enough oxygen, right? So that's angina, chest pain. So people who experience chest pain, they are sometimes prescribed aspirin to help thin the blood. Right? So aspirin, besides being a good pain medication, it also causes your blood to become thinner, which allows them to pass through the blood vessel easier because now they're narrow, right? due to the block. So you want the blood to be less thick and be able to pass through easily. That's why if you visit a dentist, sometimes the first time you visit them, they ask you to fill in like a questionnaire, and one of the questions they ask is like, "Are you on aspirin?" Right? They need to know because if you're on aspirin, your blood is thinner, and when they do the dental work, it could lead to bleeding that is difficult to clot. All right, and other thing that they might be taking is this thing called nitroglycerin. Nitroglycerin was originally they still are being used, right, as explosive. Right, these things like are used to make bombs. But somehow they found out. I don't actually know the full story, but somehow they found out maybe someone was licking the explosive or something. They found out that the nitroglycerin can be used as a vasodilator. Yeah, so it can be in the form of a pill that people put under their tongue, and then it slowly dissolves and then it opens up your blood vessel. That's what vasodilator means. Okay, so combine that with the thinner blood, it's able to restore blood flow and take care of the angina. All right. If it's a partially blocked vessel, there is a surgical option called balloon angioplasty. In angioplasty, a catheter is inserted through the femoral artery, through the leg. Okay, somewhere I can put my legs that high up. Okay, but it's through the leg, it's guided through ultrasound, and then they feed the catheter all the way to where the block is. And they would know where the block is because they would have to take what's called like an angiogram, like an X-ray after you drink some, after they inject some dye into your blood. Then they could take like an angiogram to see where the blockade occur. Right? And then they feed this catheter to the blocked location, and once it gets to that place, there is a balloon at the tip of the catheter, and then they inflate the balloon, and basically the balloon uh, opens up and flattens the plaque. It's a little bit hard to visualize. There's a video I post in the video archive, but let me try to draw this out for you. Okay, you don't have to draw this. Okay, so just watch. There is the plaque. Okay, and here comes the catheter. Okay, and then they're gonna inflate a catheter, so the catheter tip has a balloon. So the balloon is gonna basically, you know, opens up, and then it flattens the plaque, and then they deflate the balloon, and then they remove the catheter. Make sense? All right, so a couple of things about this angioplasty. Because the surface is still rough, over time, the clot will come back. And it'll just go back up. And then you might have to do another angioplasty. So that's what happens at the beginning. But nowadays, there are new technology that can leave behind like a metal stand inside your body. Okay, so they leave like a little net on the inside, and it prevents it from closing again. Right? So that's balloon angioplasty. Right? Restenosis, re-narrowing is common, about 21%, I think. But sometimes a stent is inserted into the vessel to keep it open, prevent it from collapsing. So aspirin, nitroglycerin, balloon angioplasty is for partially blocked blood vessels. If it's completely blocked, then it's going to be a full-fledged heart attack, which is called myocardial infarction. Infarction means death. Okay. So myocardial infarction is like the death of heart cells, basically, heart muscle. Right? It can happen in the brain, then it's like a infarction in the brain, right? Okay, that's a stroke. It's the same thing. Stroke and heart attack are basically the same thing. It's the blood vessels being blocked. It just happens at different location, right? So in this case, surgery is going to be needed to restore blood flow. Uh, and that's what these blood vessels are for. They would take a vessel usually from the leg, and then they create a detour, an alternate pathway for the blood to flow. So let's say there's a block here, right? Yeah, there's a block here. So the blood's going to come down from the coronary artery because of the block. This whole area is not going to get any blood supply. Right? So in coronary bypass, this vessel is not supposed to be there in the heart, okay? But they take it from the leg. You have a lot of overlapping ones in the leg. It's not going to be a problem. You take one, and then you stitch it directly to the aorta on one end, and then the other end is going to go downstream of the block, and then basically, it restore the blood flow. So in this particular diagram, this heart has a triple bypass, and there are three of these graphs that are created. All right. And that is it. That is the first lecture, everybody. Your first quiz is going to be all of lecture 1. And I don't remember off the top of my head when that is happening. But let's find out. Okay, if you go to course information, your weekly schedule, you will see that the first quiz will be January 20th. So next Monday. Okay, and it's just gonna be all of lecture 1. Right? So you have some time to study. Make sure you do the study guide, and over here in the I think it's in the studying resources. There is the study guide, and then that's the study guide answer. Now, in the study guide, actually, let me see the answer. Okay, in the study guide, yeah, there are the answers, obviously, and then there are some application questions. Okay, I'm not going to take them up today. I want you to try them out first. And I'll take it up first thing at the beginning of Wednesday's class. Okay, but please try them first. These will show up in your quizzes as well. So that's it for today. Print the notes for lecture 2 on Wednesday. And the study guide. Okay, we will be using the study guide quite extensively for lecture 2. Any questions before you go? Well, you know where to find me if you have anything later on. Enjoy the rest of your day, guys. See you all on Wednesday. Bye-bye.